Okay, in our first lesson, we left off with this slide looking at the results of Mendel's first cross between a pea plant that had purple flowers and a pea plant that had white flowers. And Mendel's interesting finding that all of the offspring had only purple flower color. Not a single one had white. And by the way, this is meant to represent a ratio. There weren't just 4 offspring plants. There were many more than that. But what it's showing you is that 4 out of 4 or 100% of the offspring had purple flower color. Now, remember that there were other easily observed traits in the pea plants besides just flower color. I'm just showing that one here as one example. But in addition to that finding, an interesting trend was seen with the other traits that are heritable. For example, when crossing a round seeded and a wrinkled seeded parent plant, all of the offspring in the F1 generation only had round seeds. When crossing a plant with yellow with green, another one that had green seeds, all of the F1 generation had yellow. And you can see in this trend here that for these parental crosses that only one of those traits showed up in the F1 generation offspring. So this led Mendel to another question. Where did the other variation of the trait go, which we now know that term for that is recessive alleles. And we'll circle back to this later, but in other words, where did the white flower trait go? Where did what was another one? The wrinkled seeds, the green seeds. Why did only one variation of the traits show up in the F1 offspring? So to get into this question, Mendel did another round of experiments. He took the F1 generation offspring, like the purple flowered pea plants, and he allowed them to pollinate each other. Through self and cross pollination, he took the F1 offspring and did another round of crosses. And here, the result was that the recessive allele came back. The trait that was hidden in the first round came back in the second. So there was a 3-to-1 ratio of the purple flowered plants compared to the white flowered plants. And he saw the same trend in the other traits, that also the one variation disappeared in the F1 generation offspring, but reappeared in the F2 generation offspring, or second filial generation of offspring, in a about 3-to-1 ratio. These are his actual data. He kept very specific notes. He had a very detailed logbook, and his experiments were quantitative in nature, so really robust data, reputable data. And so with thousands of seeds counted, seeing that 5,474 were round, 1,850 were wrinkled, I mean, that's pretty compelling data to get a really close to 3-to-1 ratio. And so all of them together are really close to that 3-to-1 ratio of one variation of the trait to the other that was hidden in the F1 generation offspring, or that didn't show up in the F1 generation offspring. So from these 2 experiments, Mendel arrived at some conclusions that comprise his model and ultimately his laws. So he developed a hypothesis to explain the 3-to-1 inheritance pattern that he observed in the F2 generation offspring through a 4-concept part model. And these concepts are still used today to explain what we know about genes and chromosomes. Let's take a look at those 4 concepts that make up Mendel's model. First concept is that alternate versions of genes account for variations in inherited characters. Here's a chromosome, one single chromosome, not duplicated, and on that chromosome, there's going to be genetic information that's carried. For example, on this chromosome is the genetic information for flower color. The specific location for that genetic information on the chromosome is called the locus. And at the locus, there's going to be an allele, or whatever version of the gene is on that chromosome. So for example, this one has the allele for purple flower, not the allele for white flower color. For the pea plants, though, an organism, a full-blown pea plant organism, would need to have 2 chromosomes for that trait, and therefore they would have 2 alleles. And this brings us to concept number 2. Mendel recognized that for each character, an organism inherits 2 alleles, one from each parent. So here we could say we have the 2 chromosomes. They're homologous chromosomes. One came from mother, one came from father, pea plant, and one carries the allele for purple while the other carries the allele for white. Now, in this scenario, we're seeing where there's 2 different alleles, but there could be the case when one has both the alleles for purple or both alleles for white flower color. When they're different, though, which was the case with Mendel's F1 generation offspring, then one was dominant and the other is recessive. So here's that term recessive that we were talking about earlier. What that means is that when you have a dominant and a recessive together, that the recessive allele is masked. It has no noticeable effect on the appearance of that organism for that trait, while the dominant allele is the one that determines the organism's appearance for that trait. So the F1 generation actually had the alleles that we just talked about, an allele for purple, an allele for white, one from each parent, but they did not express the white flower color because it's recessive to the purple flower color allele, which is dominant. The last concept of Mendel's laws, or leading to Mendel's law, the first law, is the law of segregation. And this is rooted in the process that happens during cell division, and I'll show a diagram on the next slide for this, where the 2 alleles for every heritable character or trait are going to separate or segregate during the formation of the gametes and end up in different gametes. So let's take a look at that visually, with an emphasis on the left in the purple box, the law of segregation. So we're looking at 2 traits here. We have the round versus wrinkled seeds and yellow versus green seeds. So the 2 cells at the top that are going to go through meiosis, they have 2 alleles for each of those 2 traits. So there's a chromosome that has the capital R, which is representing the dominant allele for round seeds, and another chromosome carrying the allele for wrinkled seeds, lowercase r. And then also we have another chromosome carrying the dominant allele for yellow seeds, capital Y, and another chromosome carrying the recessive allele for green seeds, lowercase y. So let's just follow this one on the left through. And so through meiosis, of course, this is an abridged version, you get the duplication of each of these chromosomes before meiosis begins. In fact, that would be in interphase. And then they come together and form the tetrad of homologous pairs. So all the chromosomes that carry the genetic information for round or wrinkled seeds are going to be part of a tetrad, and all the chromosomes that are part of the carrying the information for the color of the seed are going to form a tetrad. The tetrads separate, bringing the sister chromatids to opposite poles, and then in the next round of meiosis, sister chromatids line up, and then they get separated and ultimately form the gametes that are haploid. Each gamete, notice, has only one allele for the color and one allele for the round or wrinkledness, one letter R and one letter Y. So this is the law of segregation, that the 2 alleles that started together in the parent cell end up separating to form the gametes that have to have half the number of the normal body cell or the normal somatic cell. Now we'll come back to this same diagram when we talk about Mendel's law of independent assortment in a later lesson. But for now, we're going to leave it at this because we just want to focus on how each trait separately can lead to the appearance of the offspring, as we saw with Mendel's first and second experiments.